okay? So you have to remember a couple of basics from your prior phases of education. So you have the so-called major histocompatibility complex, and that codes for the MHC molecules or the human leukocyte antigens. We have, in broad strokes, two major classes of HLA antigens that are important for renal transplantation. The class 1 antigens, which are visually portrayed in this figure, are the HLA A, B, and C molecules, okay? And those are present on all nucleated cells, okay? Teleologically, these are tasked with presenting viral proteins of an infected cell to the cell surface, okay? That is endogenous antigens, antigens that are manufactured in the cell by the cell's protein synthetic machinery. It then binds to this cleft within the MHC class 1 molecule and then is transported to the cell surface so that an immune cell can recognize this protein and then make a decision. Do I think this is non-self and needs to be destroyed, or this is self and can be tolerated? Okay. This molecule, the mature molecule, has a variety of, let's say, segments to the mature protein, and that is also on the cell surface in association with beta-2 microglobulin, the same beta-2 microglobulin that you might measure when prognosticating in multiple myeloma, the same beta-2 microglobulin that can accumulate in dialysis patients with older dialyzers, okay? And again, this is designed to be recognized by these immune surveillance system, the cytotoxic T cell, and say, okay, this is something I'll either ignore or I will destroy, okay? Class 2 MHC or HLA molecules are comprised by the HLA-DR, DP, and DQ loci in your genome, okay? Again, A, B, and C are loci on chromosome 6 coding for these class 1 molecules. DR, DP, and DQ are also additional loci that are coding for class 2 antigens, okay? These present so-called exogenous antigens. So a professional immune presenting cell, like a dendritic cell or a B cell, those can also present antigen, can recognize antigens that are in existence outside of the cell. So for instance, a virally infected cell starts cranking out protein that is derived from the viral genome. Those proteins might accumulate in the extracellular space. Class 2 molecules can start recognizing those proteins, bind them in the binding cleft of a class 2 molecule, and then present it to other immune cells and make a decision of, okay, let's start identifying this. Let's increase in number and start going to look for this antigen expressed on other cells and start destroying them, okay? So again, class 2 molecules are present on professional antigen-presenting cells. I've enumerated several of those, okay? Even endothelial cells that are injured, so-called activated endothelial cells, also can express class 2 molecules, and that is why you can destroy an allograft, which is expressing class 2 molecules that are uncompatible or incompatible with the host, okay? You might say, well, If class 2 antigens are only present on immune cells, and I, as a transplant recipient, start producing antibody to class 2 molecules, then I should only be destroying the white cells in the allograft. But that's not the case, because the allograft endothelial cells can start expressing class 2 molecules as well, okay? Now, the co-stimulation molecules, okay, are part of that decision to say, okay, there's a protein that's bound to these binding clefts, and I can then make a decision. I can say, okay, I recognize that protein, and I can say, tolerate it or destroy it, okay? That step, that decision is based on these interactions between what we'll call co-stimulation molecules, okay? That's the next signal that is involved in, again, allo recognition, meaning recognizing non-self, or tolerance. Okay, I recognize non-self, but I'm going to say, don't do anything here. Okay, co-stimulation is an important step in that immune recognition, and is something that we modify, right? We give belatacept to block this decision to say, okay, react. Okay. Now, these class 2 molecules, which have a protein bound to them, are engaged by the T cell receptor, okay, the CD8 positive T cell. The T cell receptor, you might remember from, again, immunology, basic immunology for medical school. They would bind this HLA molecule and this protein that's bound in the binding cleft. And then these co-stimulation molecules that are present on the T cell, right? CD28 binds to these antigen presenting cell surface molecules, B71, B72. The alternate name for B71 and B72 is CD80 and E86. Again, the T cell has the CD28 that engages with these co-stimulation molecules, and then can make a decision to, again, recognize non-self and react, or recognize non-self and tolerate. You'll perhaps recognize this CTLA4 as, again, a target for belatacept. This is also a part of immune or immune immunotherapy in cancer. Okay, so these signal 2 or co-stimulation is also modified in the space of oncology in the opposite direction. So we try to modify co-stimulation and say, tolerate this, don't react. Our oncology colleagues are giving molecules that block CTLA4 or downstream steps to say, okay, I want you to react. Okay, upregulate your reactivity so that you can recognize cancer cells and destroy them. Okay, so those are some of the basic immunology that you should know that will help you make sense of things like the cross match, okay, or how does belatacept work? How does other immune tools or immunotherapy tools work in transplantation? Okay, so let's shift gears over to matching. We try to match allografts both in terms of ABO, so we look for ABO compatibility, and we also are trying to match at HLA, okay? So let's get some background behind us. The blood types they vary in the US population. The most common blood types are blood group O. Okay, if you remember, that's the universal donor. But if you're a recipient with blood group O, even though that's common, that's a disadvantage because you can only accept blood group O organs, right? You would be incompatible to all the other people expressing blood groups A or B, okay? So again, O is that universal donor, but it's bad if you're a transplant recipient, okay? Now, on the other side, AB, if you're a recipient looking for an organ, AB is rare in the population, but the beauty of being an AB recipient is you can get a blood group O kidney, an A kidney, a B kidney. You're compatible with everyone. Okay, so this is 5% of the US population. I always say I belong to this 5%. Okay, because my kids have A and B, and my wife is O, so I must have been this AB guy. Or something else is awry. Okay. All right. So wait times by blood group. Again, if you're in a blood group O or B, you're waiting on average in this area about 4 years. Okay. Notice again, B is a somewhat, it's a pretty small population. So O and B have the longest wait times. A moderate wait time. Notice if you're a blood group AB on the wait list, you can make an argument that maybe you don't even need to work on that fistula, okay? Because it looks like you might get a kidney in less than a year, okay? And there are racial differences for these blood groups, and African Americans are disadvantaged throughout nephrology, I would say, in terms of prevalence of disease, and their access to transplantation is also disadvantaged, although those disadvantages have been at least partially ameliorated. But one disadvantage is that African Americans have a greater prevalence of blood group B. And so they are disadvantaged in having to wait longer for the compatible blood groups. Okay, let's move on then to HLA matching. HLA matching that is relevant to renal transplantation when we're allocating points for the wait list are focused on A, B, and DR. If you're matching a bone marrow transplant, you try to match at even more HLA loci because there are, right? There's a HLA-C, there's DP-DQ, but historically, renal transplants are focused on these 3 loci, okay? Now, for each one of these HLA loci, remember, you have 2 alleles, one that you got from mom, one that you got from dad, okay? And so you have 6 different HLA antigens, right, or alleles, coding for antigens that you express on your, remember, class 1 molecules, A and B, are on all nucleated cells. They recognize endogenous antigens, right? Any nucleated cell should have the ability to say, I'm virally infected. I better express this viral protein on my cell surface. So all nucleated cells have HLA class 1 expression. Now, the DR is, again, the class 2 HLA molecule that is present on these antigen-presenting cells, like B cells, dendritic cells, macrophages, okay? Again, we are focused on these 6 alleles when we are deciding or making statements about rightness of fit or compatibility, okay? Now, there is a difference in outcome, but that difference is modest except at the extreme of a 0 antigen mismatched kidney. So if you have a kidney which matches on all 6 alleles, okay, a donor that expresses, for instance, a donor which is A2, A, 7, I'm just making things up, B27, B12, dot, 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 right, by my DR. And I have a recipient that has these same HLA molecules that they express. This would be considered a zero antigen mismatched kidney. Okay. Another way you can be zero antigen mismatched is if the recipient has B27 and also got from mom and dad B27. Okay. So even though this individual does not have a B12, I'm not having incompatibility because, again, this recipient is sharing this B27. Okay. So actually, it's in the inverse. Excuse me. The donor B27. Okay. And I might have a B12 in my recipient. Excuse me. Remember again, I am looking for incompatibilities, right? Things on the donor that I have never seen. Because the donor has two B27s, this would also be a zero antigen mismatch kidney. Sorry for that misstep, okay? Inverse that. All right, so this would also be considered a zero antigen mismatch. Those have detectably better renal outcomes at 5 years compared to 6 antigen mismatch kidneys, where I'm mismatched at every single one of those. But notice that the difference is not huge, right? 10% difference at 5-year allograft outcomes, okay? But this is enough, this improved outcome is enough to say, if I have a zero antigen mismatch kidney, I'm going to ship that kidney further in this allocation schema to make sure it gets to the best matched recipient, okay? Because presumably that kidney will last longer, it will have more good, right? It will keep a patient off dialysis longer, and therefore will translate into maximized good, okay? There are other things to be aware of. When you mismatch at DR, those have detectably worse allograft survival. When you mismatch at B, those also have detectably worse allograft survival. And so these are relevant for our point system, okay? You get points as you're waiting on a wait list. And those points vary when a kidney becomes available, right? You have your points, 1 point per year of wait, right? We'll go to the point system later, but I'll just give you a prelude to that conversation. So if I'm waiting 3.5 years, I have 3.5 points, okay? And if I also am matching a prospective donor at DR, I temporarily get extra points, okay? Because it turns out that DR affects outcomes. And so in order to maximize the good, transiently, you give extra points to people on the top of the wait lists in order to maximize the good of this donor recipient pair, okay? That used to be the case for B as well. However, that was removed because it clearly disadvantaged patients of African American descent. Okay, that's different from blood group B. There's another point we'll make later on. At the HLA-B locus, African Americans have greater variability at HLA-B, and so they're less likely to match prospective donors at the B locus. Okay, and so the unintended consequence was, well, they were not getting those points that non-Black people were getting. And so that created disadvantage. So we said, okay, even though we're trying to maximize good, we should not penalize specific, especially socioeconomically disadvantaged groups. Okay. All right. There is this historically significant concept these cross-reactive groups, okay, it describes portions of HLA molecules which are called public epitopes, okay? So some of these class 1 molecules, which are coded again by alleles on chromosome 6, some of them have common features, okay? There may be an area on this A2 segment of this class 1 molecule. Let's say a B27 molecule. A portion of this B27 molecule, let's say in this A2 region, has commonalities with the A2 region of a B2 molecule, okay? That would be called a public epitope. And the problem is, if the recipient starts producing an antibody to this public epitope, they are now incompatible with more than one HLA molecule. They will be incompatible to a B27 and a B2, okay? These cross-reactive groups describe these overlaps between different HLA molecules because they share public epitopes, okay? And that's why, again, a prospective recipient who has only a few HLA antibodies translates into an incompatibility with a wide swath of prospective donors. Again, you may only produce a few alloantibodies, but if those alloantibodies are directed to public epitopes, you become incompatible with like 70% of the donors. Okay? That is a problem. And that's when sometimes we'll see donors who have incompatibility. Panel reactive antibody is that description of incompatibility, which we'll talk about. Panel reactive antibody to 100% of donors. And that's often because they're producing alloantibodies to multiple public epitopes. Okay? Moving on to the crossmatch. So you have a recipient and a donor pair that become available or become, the computer model says this potentially is compatible, okay? Now we have to go and check for that, okay? There's the virtual crossmatch that's done. Here are the actual experiments that are done in the middle of the night in order to ensure that there is compatibility between a donor and recipient pair, okay? There's a T cell crossmatch and a B cell crossmatch, okay? I can use peripheral blood from the donor, right? The donor becomes available. A donor has brain death. I collect blood from that donor. I can then recover T cells and B cells from that donor blood. If you recall, B cells are much fewer in circulation than T cells. So in a more ideal scenario, I have some donor lymph tissue, a lymph node or a spleen, in order to get more B cells, but I don't have to have that. Okay, I can get it from donor blood, both T cells and B cells. Okay, I use the crossmatch to look for incompatibilities, but let's go through some of the possibilities here, okay? We'll call them donor T cells, donor B cells. Okay, we said donor T cells, just like any other nucleated cell, expresses class 1 molecules on its cell surface. They express A and B. They also express C, which is class 1, but we don't pay attention to those for renal transplantation. B cells from the donor are just like any other nucleated cells. They're going to express class 1, But if you recall, they're also considered professional antigen presenting cells, right? B cells also express class 2 molecules, okay? So they have class 2 molecules present too, okay? The DRDP, all right? DQ. All right, so I take donor cells, I mix recipient serum, okay? That recipient serum, again, this is from the recipient, may have preformed antibody, okay, circulating. These, again, are not cells. This is serum, okay? So those preformed antibodies might react, okay? If they, for instance, if I form a preformed antibody to A2 and the donor expresses A2, that would be a positive cross-match. Or if I have an antibody to, I don't know, A7, but it shares a public epitope with A2 on the donor, I would also have a binding of that recipient-derived alloantibody to the donor-derived T cell, okay? I would also have, if this was, again, an antibody to class 1 molecules, right? And those class 1 molecules were expressed on the recipient T cell and B cell, my cross match would be positive for the T cell and the B cell, right? I can surmise, if I get a result like this, that my recipient is producing an antibody to a class 1 molecule if both the T cell cross match and the B cell cross match are positive, okay? Now, if I have a negative cross match to the T cell, but a positive cross match to the B cell, I'm going to presume that this preformed antibody is actually binding a class 2 molecule, okay? Not a class 1 molecule, okay? So this shows up as negative, the T cell cross match, the B cell cross match comes out positive, okay? There is one more twist here. It turns out that B cells express more class 1 molecules than T cells. So it's still theoretically possible that one other explanation for a positive cross match for B cells and a negative cross match for T cells is an antibody to a weak antibody to class 1 molecules. Okay, so those are your if this, then that. Okay, if you find this result, then you can presume it's a class 1 molecule, an antibody to class 1, an antibody to class 2, or a weak antibody to class 1. Okay, those are your possible explanations here. Okay, again, that's all summarized here. Your T cell cross match is used to detect class 1 HLA molecules. Your B cell cross match can detect both antibodies to class 1 and class 2. And again, the B cell cross match may be more sensitive for weak antibodies to class 1 HLA. Okay, and that's because of the greater expression of HLA on B cells. Okay, now the B cell is more likely, here this statement is basically saying it's more likely to have, let's call it false positive antibodies. I guess I should tell you, the way that we signal a positive result is we run recipient serum, donor T and B cells, and then we also mix in a anti-human globulin, something that binds immunoglobulin, okay, that has a fluorescent tag bound to it, and we run it through a flow cytometer. okay? That lighting up is detected as a positive result, okay? So you can also have some false positives that can arise because if you recall, B cells can bind immunoglobulin because B cells have FC receptors, right? I can bind the heavy chain, that FC refers to the constant domain of immunoglobulin, through FC receptors on B cells. Okay, sometimes this immunoglobulin, this allo antibody, let's use that term. Allo refers to antibodies to HLA molecules that are non-self. Allo antibody is actually stuck to the B cell through the interaction with the FC receptor. So this is one way in which B cells can result in a sort of a false positive. Translating that to patient care, if I have a B cell crossmatch that is weakly positive, that is not an absolute contraindication to transplant. You may still say, okay, I think that this is an HLA molecule, a weak HLA to class 1, and this is the best chance that this recipient is going to get. We just have to be heavy-handed with our thymo and our calcineurin inhibitor levels and we're not going to do a steroid-free protocol in this individual, okay? Or it may be surmised that this might be a false positive B cell cross-match that's positive, okay? And so we can still go. This is an older assay. We don't do this anymore at our institution. We did it when I first got here, and it's still relevant if you're considering something like HLA incompatible transplants, right? Doing a transplant, even though you may know that the individual has indeed an HLA antibody that is binding the donor HLA. In some centers, not in ours, you might still push that envelope and say, okay, this is the best chance that this patient is going to get. We're going to try and do plasmapheresis and IVIG and thymo or Campath, in order to surmount this incompatibility, okay? But even these centers who do HLA incompatible transplantations would not do a HLA incompatible transplant if this cross-match was positive for the cytotoxicity cross-match assay, okay? What is the cytotoxicity cross-match? Remember, again, this flow cross-match, this is a flow cross-match where I mix T cells and B cells, recipient serum, an antibody to human Ig, that anti-human globulin, which has a fluorescent tag. This is a flow cross-match, okay? Even weak binding by these antibodies to donor cells will show up as a positive signal, okay? A cytotoxicity cross-match, on the other hand, okay? Let's just focus on the left side. Okay, cytotoxicity cross-match takes the donor T cells and B cells, mixes in recipient serum, okay, adds rabbit complement, okay, okay, and then looks to see whether you have so much antibody that's binding this alloantigen that it can activate complement through the classical pathway of complement activation and cause cytotoxicity of the cell, okay, the cell lysis, okay? There's some nuance here, even in cytotoxicity cross matches. There's an effort to increase sensitivity by adding anti-human globulin. There's also some pre-treatment to wash away non-specifically bound antibody. There's even treatments that are used to remove IgM that may be bound, so-called DTT modification. All of that, since I used to have all this information in here, but since cytotoxicity cross-match is becoming a relic of the past, I'm not belaboring those points, okay? Only that this is a signal that means there's a lot of incompatibility here, okay? And so even if you're doing higher risk transplantations, if they have a cytotoxicity cross-match that's positive, no center would try and surmount that, okay? They would be worried about, this is how you can get hyperacute rejections in the OR. The kidney starts looking dusky, and you lose the kidney within hours, okay? This is, again, less sensitive than the flow cytometry cross-match. Flow cytometry cross-match is the more sensitive test, okay? So here I gave you the background of what's the difference between T and B cells. I've discussed the cytotoxicity cross-match, and then flow cytometry cross-match is, again, does not require complement activation. It uses a fluorescent tag and a flow cytometer in order to look for a positive signal, okay? And is less specific, but more sensitive, okay? We also do virtual cross matches. How does that work? Well, recipients who are on the waiting list have their blood sampled roughly quarterly, okay? They're going to dialysis. The transplant center asks the dialysis units, we need some blood from this recipient. Please draw blood at dialysis and send it to us, okay? Roughly quarterly. What are we doing there? Recipients on the wait list may get transfused or may have a pregnancy, and those can be sensitizing events that make that recipient start to produce alloantibodies, okay? Those are the two main sensitizing events. That's why women who have had multiple pregnancies, especially multiple pregnancies with more than one partner, can be very sensitized, right? Because they have a baby. That baby has HLA molecules that are expressed by mom, half of them, but also HLA molecules that are expressed by dad. So if you, as a woman, have a fetus with multiple HLA molecules from dad, and then particularly if you've been pregnant with partners, different partners, you can start producing more and more alloantibodies. So those are things that can cause sensitization in patients who are on the wait list. So the transplant center samples blood from these prospective recipients and says, okay, this recipient is now producing an A2. We better put that in our library. So when a donor with an A2 becomes available, we're not going to waste any time. We're not even though this patient may be at the top of the waiting list by virtue of wait time, why would we bring them in? That's just leading them on. They are not going to be compatible because this virtual cross-match reveals this patient to be incompatible, okay? So that's what is done when a patient gets called in. We've done the virtual cross-match, and they are presumably compatible. That doesn't take the place of a T cell cross-match and a B cell flow cross-match. Those are done when the patient arrives, okay? The virtual cross-match, again, allows us to, be more efficient in our organ allocation process and in the night of the transplantation in evaluating the right donors, okay? The way in which we identify, in order to do the virtual cross-match, we need to detect specific antibodies that are circulating. Notice that these flow cross-matches do not tell you the specific identity. When you have a positive crossmatch, a positive T cell crossmatch, or a positive B cell crossmatch, you don't know specifically what is the incompatibility. Maybe you can say in broad strokes, they're incompatible at some A2 HLA a class 1 HLA molecule. Why? Because the T cell crossmatch is positive and the B cell crossmatch is positive, right? But you can't say they are incompatible because they have an A2 antibody or a B27 antibody. Okay, you can't say that based on the flow crossmatch. It's giving you limited information. The way in which you create a virtual crossmatch is you have to know that recipient has not just, preformed antibody to some A1 molecules, a class 1 molecules. They have to say this individual has an anti-A2 alloantibody. That is done, okay, by running Luminex studies. Okay, that's what most centers do. There's also an ELISA-based assay where you can know the identity of these antibodies. Most centers use this Luminex technology. What is it? Well, it's a technology that relies on binding known HLA antigens to beads, okay, plastic beads that have known HLA antigens on them. Instead of using, again, a T cell in order to mix with recipient serum, I now have a bead with an A2 on it that I mix with recipient serum, okay? And when that creates a positive signal, which I'll just use as this shorthand, I now know that the preformed antibody that this potential recipient is producing is directed to an A2 bead, meaning this is an anti-A2 antibody, okay? When I know that this antibody, or when I see a positive signal to multiple beads, okay, let's say that share a public epitope, you might even make generalizations that this antibody is most likely directed to a shared public epitope. For instance, I use the example of some side chain in this A2 region that is shared across multiple HLA molecules. You can use the output of this Luminex assay to make assumptions that probably they have an antibody to a public epitope based on the pattern that I'm seeing here. Okay? So that is the Luminex study. What do we measure at the bedside when you're checking you're checking for donor specific antibody? What you're really asking for is a Luminex study looking for all of the allo antibodies, that the recipient may be producing. Okay, so if the recipient is producing an A2, a B27 allo antibody, okay, and maybe even a DP, 5, okay, the recipient has all of these allo antibodies that you detect by a Luminex study, okay, but only A2 might be donor specific. It may be a DSA, because even though the recipient produces these others, the donor kidney does not express a B27. The donor kidney may only express A2. And so this is the only donor specific antibody. Okay. So if you look at the output report, when you send the Luminex study, it says the recipient is producing these 10 different antibodies. Okay. And it may even say that calculates to a panel reactive antibody of 70%. What does panel reactive antibody mean? It means if this recipient does indeed have these 10 antibodies, this recipient would be incompatible with 50% of the donors. That's your panel reactive antibody. It just tells you how generally sensitized is this individual compared to the donor pool. Panel reactive antibody refers to the percentage of prospective donors in our pool that this individual appears to be incompatible with. It's bad to have a high PRA. If you're producing no alloantibody, your PRA by definition will be 0%, right? It means you're incompatible with no one, right? But once you start producing alloantibodies, you then start having incompatibility, and then you will be described as having a panel reactive antibody to, meaning incompatibility to 30%, 90% of the prospective donor, or in some cases 100% of the prospective donor. You can have a panel reactive antibody of 100%. Okay? Again, when you get the output of your Luminex study, your HLA lab will say this individual is producing these 10 antibodies, but only 2 or 1 is donor specific, right? Meaning this is the incompatibility between this recipient and the kidney they've received, or their kidney they're about to receive, which they won't receive because the virtual cross match is positive, right? I'm producing an anti-A, the prospective donor has an anti-A2, the prospective donor has an A2, the virtual cross match is positive, leave the patient at home. Don't even bring this person in. Okay?
Dr. Sam, to create the virtual cross match. If that's negative, because that's done quarterly at best, then
Well, the cross match can't be done quarterly.
sorry, the Luminex, sorry,
Yeah, you could do the Luminex, the anti-HLA antibody assessment. The Luminex study is done quarterly-ish.
and then we use that output to do the virtual cross match when a potential donor comes along. If that's negative, then the patient's brought in and does a T cell, B cell cross match.
Correct, which is done by flow cytometry.
And that's that day of test.
That's the day of, the night of.
And then if that's
Takes about 4 hours.
if that's great, then we potentially proceed with it.
Correct.
Okay.
Dr. Singh, could you explain why the patient was DSA only to A2, not to the B27 in your example?
Well, I mean,
In your example, like you said, the DSA was only for A2.
yeah, because I need a little more room. Okay. Again, let's say okay, again, the recipient has an allo antibodies. So these are the allo antibodies that are produced by the recipient. And let's say again that the recipient is producing an antibody to A2, right? An A2 antibody. They are producing a B27 antibody, okay? And a DP5 antibody, okay? These will show up on your Luminex study. The Luminex study will say, this recipient has an A2, B27, and a DP5, preformed antibody, okay? And that might also translate, they'll say, okay, this has, well, they'll give you actually 2 PRAs. They'll give you a class 1 PRA and a class 2 PRA, right? Remember, A2 and B27 are antibodies to class 1, okay? So let's do that. Your report will say, this results, okay, they will say, this results in a panel reactive antibody of 15%. Okay, it says, by virtue of the fact that this recipient produces an A2 and a B27, they're incompatible with about 15% of the donor pool at class 1 HLA. Okay, and they'll say your PRA for class 2 is going to be 30%. Okay, something like that. That you'll get two numbers out, PRA class 1, PRA class 2. Okay, and then he'll say, but only A2 is donor specific. That's your DSA, your donor specific antibody. Why? Because the donor, okay, has the donor kidney expresses A2 on the cell surface, right? It has an A2, A7, right? Remember, again, each of us is expressing two different HLA at each locus, right? Because we got one from mom, one from dad. And then has a B4, B, 50, a DP, well, we do DR, but you also do have DP. Let's do DP6, DP, 8, right? This is the donor HLA molecules that are expressed on the allograft, right? So even though this recipient is producing a B27, that's not donor specific, okay? It's important. It tells me how immunologically revved up this individual is. So we use, even if you have preformed antibody that is not donor specific, we pay attention to that. Why? Because it turns out that as you start recognizing non-self, okay, you're more likely to recognize other non-self, right? You're getting revved up. So even though I may have alloantibodies preformed that are compatible with this donor, if I'm making lots of alloantibody and I have a high PRA, we say, give this patient a lot of immunosuppression. Even though these two individuals are compatible, we worry about, let's say, those spillover effects or those, this patient who has this revved up immune system that is already recognizing non-self is only a few steps away of creating a donor-specific allo response. That's why, again, you might say, okay, well, these people are compatible. Why do we have to give more thymo to this patient just because they're producing a lot of alloantibody? Again, recognizing non-self is only a few steps away of saying, I recognize you, not just non-self broadly speaking. I recognize this allograft that is coming to me. Okay?
In real life, we're looking for DR, not DQ. That's what it said in
Well, it is true that in matching, in organ allocation, okay, you only look at A in kidneys, A, B, and DR, okay? But when you get the report from our Luminex study, it gives you the preformed antibody to A, B, it'll give you C, it'll give you DP, DR, DQ, all of that is given. And all of those are important, even though they're not important for allocating organs and giving points to people. It is important in the individual patient and their allograft dysfunction, right? Even though, again, I'm not paying attention to DQ in allocating points. If you have allograft dysfunction, I better pay attention to an incompatibility at DQ, right? Okay. Let's do a few minutes of kidney allocation and then we'll end it there. Okay. So what is the infrastructure of kidney allocation? It's evolved quite a bit. there are these organ procurement organizations, and it may be evolving even more. There's been lately in the press, a lot of criticisms of this existing system, which is based on Medicare contracting, or the government, federal government contracting with these non-governmental entities called organ procurement organizations to identify donors, identify recipients, and then match people efficiently. Okay, keep track of points accurately. People have done exposés to show, there are people who have been falling through the cracks, and there's all these records that are based on paper, and it's not efficient, and so this may be upended soon. Okay, there are 58 organ procurement organizations in the US. There are 3 in Texas. They're the so-called donor regions. These 58 OPOs are broken into 11 donor region. Our region in Texas, I think, also includes Oklahoma. And then there are smaller states that have multiple states in their donor region. Yeah, Texas and Oklahoma, 1 donor region. And these organ procurement organizations manage the donor pool in this so-called donor service area, a DSA, different DSA, okay, than the Luminex output of DSA. Again, these are contracted to increase organ donation, so they actually do outreach and try and get people to sign up to be organ donors. They will also maintain the match list, and then they will arrange transportation, recovery of organs, organ procurements. Okay. These are 2 scores that you have to understand. So there's an effort to pair good quality kidneys with healthy recipients, and that's this EPTS and KDPI score. Okay. Before, there was no effort to do that. Not like 50 years ago, like 15 years ago. If you were a 78-year-old, you could theoretically get a kidney from, a 15-year-old or a 20-year-old kid, right, who died in some trauma accident, right? And so the thought was, well, that 70-year-old is going to die with a creatinine of 0.8. And that would have been, a good that could have served a healthier person for longer, right? And so that's when, about 15 years ago now, there was this revision in organ allocation, and it created this estimated post-transplant survival. So that 77-year-old will have a low estimated post-transplant survival, right? It will have an EPTS score, right, that was on the high side. High numbers are bad. Okay, so high EPTS scores are bad. Low EPTS scores are good. Okay, so a young IgA patient who has ESRD at the age of 30, or a PKD patients, those are generally more healthy patients as well, they will have lower EPTS scores. Okay, so what are the things that factor in? Age, time on dialysis, presence of diabetes, and a history of prior solid organ transplantation. Those are the things that go into an EPTS score. Notice that this is a very coarse estimate of survival, right? You might have some early bridging fibrosis in your liver, right, with your history of Hep C. That's not captured. You may have had a amputation from PVD. That is not captured as long as you're not diabetic. Okay, so this is a coarse score. When this was rolled out, there was an effort to balance simplicity with accuracy, but it's absolutely true that the EPTS score is a flawed instrument. Okay, it's one step in the right direction, but it is not, it's not a perfect estimate of matching between a good kidney and a good recipient. Okay, now on the kidney side, this is the recipient side, EPTS score. On the kidney side, you have the KDPI, okay, KDPI, which is based on a KDRI, okay? They're basically the same, okay? The kidney donor risk index, okay, is a score essentially that you get based on a number of factors in the kidney attributes. So donor age, black versus non-black race, creatinine, hypertension or diabetes in the donor. If you have a cerebral vascular accident, you might ask yourself again, why prior solid organ transplant? Is that in your EPTS score? There were multiple prospective pieces of the EPTS score. These were the attributes that had the greatest predictor for survival, okay? So that's why arbitrarily, this was just statistics. They just looked at outcomes based on recipient attributes, and they said, okay, which ones had the greatest p-value, right? They had the greatest effect size on predicting outcomes. Prior solid organ transplant was one. If a donor died from a CVA, that also predicted worse outcomes for the recipient. Height and weight. Hep C status is no longer, I got to update this. Hep C status is taken out. Okay, this was a very recent change, I'd say within the last year. And then the donor after circulatory death, we'll talk about that on another day. Sometimes, again, we wait for the heartbeat to stop in the donor before procuring organs. Those were all attributes that feed into the kidney donor risk index. Okay, the KDPI takes that kidney donor risk index and just compares that to a reference population, okay, in order to get the KDPI. This is the number we use, KDPI. KDRI is just generated from all these attributes. It translates to a KDPI, okay? Higher KDPI is bad, right? Bad quality organ or worse, okay? These two, again, are limited in its ability to predict the quality of the organ, okay? We talked about panel reactive antibody already, so we don't have to go through that. It can be, again, calculated from your Luminex assay and just knowing something about your donor pool, right? You have to know what proportion of the donors here in Texas in this donor service area have A2, right, in order to calculate a panel reactive antibody, okay? How do we pair kidney donors to recipients? We'll end probably here, okay? We won't go into wait listing. We're trying to balance utility and access. Used to be it was just all about access, right? We were not trying to maximize a good by pairing good quality kidneys with young patients. This is the broad strokes of the organ allocation system, okay? Donor kidneys, remember, low KDPI is good, okay? Those are healthier kidneys. They get allocated preferentially to highly sensitized candidates. Why? Because they just don't have many options, right? So what is the definition of a highly sensitized candidate? It's it's driven by the panel reactive antibody. So those patients with PRAs of 95%, those would be considered highly sensitized, okay? Those who have zero antigen mismatches, remember we said there's a survival advantage when you match, that well, 75% 5-year survival versus the 65% 5-year survival. So even if you're an old patient, if you get, a kidney that's a zero antigen mismatch, then you can jump to the front of the line, even though you might have an EPTS score that's relatively high, okay? Or if you have low EPTS scores, right? You can have access to these low KDPI kidneys, okay? So those three groups, highly sensitized, zero antigen mismatches, or the relatively healthy EPTS score patients, okay? But again, that does not capture it perfectly, okay? EPTS scores are not used for allocating higher KDPI kidneys. It's really just focused on taking these lower KDPI kidneys and matching them with younger patients, okay? These KDPIs, that, remember, that's the donor attributes, greater than 20 but less than 35, are preferentially offered to highly sensitized kidneys and zero antigen mismatches and pediatric candidates. KDPI kidneys that are greater than 85%, those are your lesser quality kidneys, right? Older donors or a donor with diabetes. Those could drive the KDPI score up, okay? Those can only be allocated to those individuals who have consented to take lesser quality kidneys. Why would you do that? Well, the competition for those kidneys is less. And if you want to minimize time on dialysis, you would consent for higher KDPI kidneys. If you were a young 30-year-old with IgA nephropathy, we'll tell you, please don't accept high KDPI kidneys. You can, but please don't. Why? Because you're likely to outlive that kidney, get back on the transplant list, and then you'll be sensitized now. And then going to round 2 would be disadvantageous, okay? But that 70-year-old, you would say, please do consider high KDPI kidneys, because you would, statistically speaking, do better with a higher KDPI kidney than waiting on dialysis longer to compete for all of these other kidneys, okay? Candidates with high PRAs, defined as very high PRAs, 98%, they get to see every kidney that gets on the radar. So if I have a 98% calculated PRA, if a kidney becomes available in Washington State, they'll look at me as a potential recipient of that kidney from Washington State. Otherwise, you're just looking at the local recipient pool when you're evaluating potential candidates to bring into the hospital, right? I will ship that kidney from Washington State to me in Texas if I have greater points than that person who's closer, okay? Again, if there's a zero antigen mismatch kidney, we also ship kidneys across the nation in order to make those well-matched kidney recipient pairs. And then there's this proximity to the donor location that also affects allocation. Here we'll talk about in a subsequent discussion. We'll pick this up next week. There's this you get points for being close to the donor, right? The donor hospital within a 250 mile radius, you get graduated more points. So you can see that the points are, there's a static feature. How many years have I been on the wait list? But then there's this dynamic feature to it. How close am I to this specific kidney? Do I share DR commonality? So I can go, my points can kind of go ebb and flow by a certain number based on some attributes that become available when the kidney becomes available. Correct.
The points are equal or someone's barely higher in numbers than the person at the hospital, would they favor, given that the person's at the hospital, to give them that organ or still send it elsewhere?
Yeah, here, I guess the points vary up to 0 to 2 points. Okay. It's not, there's some complexity to it beyond just, is it just a 250 mile radius, 0 versus 250. There's some complexity to it that, more features are factor into your points if you're outside a certain radius. So there's complexity to it. I'll just put it that way. And it's probably not important to know all of that nuance, because again, that's just what's driving the points. You know, a person in the OPO, a contractor, is doing all these math, presumably correctly, and then calls the surgeon and says, "Hey, it looks like you guys have a recipient in your transplant program that is at the top of the wait list for this kidney." Okay, and then well, based on the driven by the wait list points, right? Because we're essentially the same location, so we don't really get proximity points, except theoretically if the donor's in the same hospital, right? But yeah, the main thing that drives whether a kidney goes to Parkland or here is at the top of your wait list, who has the most points. Okay. And then there's also some schema for paybacks that also factor in. If someone passes on a kidney, and then there's another sort of layer of complexity there too. There's a system of paybacks. And then also you can refuse. Our UT surgeon may say, I don't want that kidney. Okay, my person is too healthy and will do well on the wait list than taking a risk on this kidney. Whereas the person at Parkland may say, it's now or never for my patient. So it may have allocated first to UT, but then the UT surgeon passes and then it goes to Parkland, or vice versa, depending on the attributes of the prospective donor. So there is clinical decision-making that's made the night of. Sometimes. Yeah, you get time zero biopsies of the donor kidney. Those have uncertain value.